The cell membrane, sometimes called the plasma membrane, is the boundary between a cell and its surroundings. A key feature of the cell membrane is differential permeability. This means that it allows some substances to pass through while keeping others out. Consider a fish net as an analogy of differential permeability. The net has holes that allow water and other substances in the water to pass through freely, but not the fish. Depending upon their size, some kinds of fish might pass through the holes in the net, while others are caught. However, the control of movement of substances across a cell membrane is more than just about molecular size. It also depends on the polarity of molecules. So, what is polarity? In a molecule, atoms are linked together by covalent bonds in which they share electrons. However, in some molecules, the electrons are not shared absolutely equally. These molecules, which have an unequal distribution of charge, are said to be polar. For example, in a water molecule, two hydrogen atoms each share a pair of electrons with an oxygen atom, forming a molecule with the formula H2O. Notice that the oxygen atom has 8 protons in its nucleus, and each hydrogen atom has 1 proton in its nucleus. Oxygen has a stronger ability to attract the electron towards its nucleus. In other words, oxygen has the electrons around its nucleus more often than the hydrogen does. You may imagine this as in a rug and tug game. When one side applies a greater pulling force on the rope, the towel in the rope is pulled towards that side. This produces a partial negative charge at the oxygen end and a partial positive charge at the hydrogen end of the molecule. Polar molecules are hydrophilic, meaning water-loving, and they tend to be soluble in water. By contrast, a non-polar molecule does not have charged ends. They are not attracted to water and are said to be hydrophobic. With this background knowledge, let's now look at the main component of the cell membrane, phospholipids. A phospholipid has a polar head that contains a phosphate group and 2 non-polar tails made up of fatty acids. In the cell membrane, phospholipids are arranged in a bilayer. The fatty acid tails point inwards, facing each other and forming a non-polar hydrophobic interior. The polar heads face the aqueous environment found inside and outside the cell. The phospholipid bilayer is permeable to non-polar molecules. The respiratory gases, oxygen and carbon dioxide, are non-polar and small enough to diffuse across the cell membrane directly between the phospholipid molecules. The rate at which this diffusion takes place largely depends on the concentration gradient across the membrane. How about polar molecules? The phospholipid bilayer is impermeable to ions and polar molecules such as water, glucose and amino acids. Ions, even though small in size, cannot move through the phospholipid bilayer, as they are repelled by the non-polar interior. Polar molecules and ions can only cross the cell membrane through the channel proteins or carrier proteins. Cell membranes contain many different types of channel proteins and carrier proteins, each type allowing only a particular kind of polar molecule or ion to pass through it. The number of these proteins available in the membrane affects the rate of transport of the polar molecules or ions. In this way, The cell membrane controls the movement of polar molecules across it. By restricting the movement of polar molecules across it while allowing non-polar molecules to pass through freely, the cell membrane is thus described as differentially permeable.